Thanks a lot, Rosalind, for the introduction. So what I'm going to present you here today is really the results of an interdisciplinary project where we combined mathematical modeling, this is my expertise, with experimental assays, which is not my expertise, even though all the most of the experiments I'm going to show you were done by myself under very close supervision by biologists. But the sort of topic we investigated was how competitive dynamics under bacterial range expansion or biofilm formation takes place. And in particular, we focused on the role the so-called founder cells, those are the cells in the inoculum of a biofilm, what role they play. In the title, I've also mentioned in the abstract as well, spatial heterogeneities. I most likely won't have the time to talk about this. But yeah, so I guess most of you in the audience know what the biofilm is. If you don't worry about it. All you need to know is that biofilm are those dense collectives of cells that basically live in an extracellular matrix that they produce themselves. On top here, you can see an example of a biofilm that was grown on an agar plate in the lab. It's Bacillus subtilis. And essentially many different bacteria species form biofilms, and therefore they occur in many different environments. From a human perspective, they can have both positive and negative effects. For example, biofilms are quite essential for the functioning of the human gastrointestinal tract. Biofilms are also used in wastewater treatment, for example. But on the other hand, biofilms are often associated with antibiotic-resistant infections, and they also cause the fouling of medical devices, but also industrial devices. But that's just a very short list of examples. One particular example that motivated us to study competition within biofilms was that of the bacterium Bacillus subtilis, which, among other places, lives in the soil. And in the soil, Bacillus subtilis can form biofilms on the roots of plants. This is what you can see on the bottom image here, where in purple you can see the root of a plant, and in green you can see a biofilm that has formed around it. And now what's quite important here is that some strains of Bacillus subtilis, but not all of them, can actually enter a symbiotic relationship with the plant, meaning that the biofilm, the bacteria, can actually protect the plant from plant pathogens and also supply them with nutrients, in particular fixed nitrogen. Now, this makes those strains of Bacillus subtilis ideal candidates to be used as biocontrol agents, or so biofertilizers or biopesticides as alternatives to currently used fertilizers. But obviously what's quite important here is that if you're producing, if you're a company selling such a biocontrol agent, for that agent to be successful, it not only needs to be able to complete the intended task when forming a biofilm on the root, but it actually needs to be able to form a biofilm, meaning that it needs to be able to establish itself in the community or even outcompete other bacteria, and in particular other Bacillus subtilis strains that are already present in the soil. And this really motivated us to study competitive dynamics. Now, what's known is that in Bacillus subtilis and other bacteria species as well, competition is underpinned by a mechanism that's called kin discrimination. So essentially, a cell can detect the neighboring cells and decide whether to launch antagonistic actions or not, depending on whether those neighboring cells are detected to be quite similar, kin, or not similar, non-kin. And many of those mechanisms, not all of them, but many, really depend on two different strains, two different cells being in contact with each other. And this really motivated us to, in the attempt to explore competition dynamics, this really prompted us to take a step back and first focus on the spatial structure in biofilms. So what you can see in those two images here are two biofilms that comprise two different strains. And actually those strains produce a fluorescent protein, so that's visible. So that's the color you can see. And you can already see by comparing these two images that the spatial structure or the distribution of the two different strains can be quite different among different biofilms. So here we have clear spatial segregation, whereas here it's a much more homogeneous mix than compared with the bottom one. Now, quite importantly, the green strain up here and the green strain here, and same for the purple, those are the same strains. So those two biofilms are actually quite similar in terms of, well, they have the same species composition, the same strain composition. I'm going to tell you in a second what the actual difference between those two images is. Now, just before that, let me mention that the best way to study how those spatial dynamics work is to look at so-called isogenic strain pairs. So strains that are identical apart from the fluorescent protein that they produce. And so this is really a choice that we made to focus on the spatial dynamics, because then by design, we include any other competitive interactions, so in particular antagonistic interactions. But obviously this is not really realistic when it comes to applications, so this is really just a first step. But we believe that to really understand how antagonistic interactions work, we first need to understand how spatial structure like this emerges, and this is something that's best studied using isogenic strains. We were not the first group to ask this question, and in particular, what has been found in the past is that the density of the founder cells, so essentially the density of the cells in the inoculum, they have a significant impact on spatial segregation dynamics in biofilms. So the difference between those two images is that the top image is a biofilm that was inoculated using a high founder density, and the outcome is a roughly homogeneous mix, whereas the lower image is a biofilm that was inoculated using a low density of founder cells, and the outcome is spatial segregation. This has been observed across many different species, not just Bacillus subtilis. This is a non-exhaustive list of papers of groups that have reported this phenomenon. But essentially at least we were not aware of any paper that really was able to answer the question why that difference in the founder density leads to those different observations. And so really the question we wanted to address was how this spatial structure, this spatial distribution structure arises in the first place. And so like I've said in the beginning, we combined a mathematical and an experimental approach to study this. Very briefly, the experimental setup worked as follows. We used initially identical strains that only differed in the fluorescent protein that they expressed. We always mixed them at a 1-to-1 ratio at a normalized OD, so always same density. And then we put an inoculum on an agar plate, incubated that agar plate, and examined the outcome. Now, we also, to come back to that question of the founder density, obviously wanted to do this over a whole range of inocular densities, so we always diluted by a factor of 10 to get a whole range of different founder densities. Now, if you know something about Bacillus subtilis, you'll immediately recognize the strain here that we've used. 3610 is somewhat one of the standard lab strains used in Bacillus subtilis research. But we also used a second strain, which we called 6153, which is a strain that we isolated a few years ago through a public engagement activity from a sample of garden soil in a small village very close to Dundee in Scotland. From a mathematical point of view, we used the PDE model, and it was really a phenomenological model that brought down the processes of biofilm formation to really the most simplest dynamics, meaning local growth and spatial spread. So essentially, we've got just two equations, one for each strain. So we just consider pairwise combinations here. And already you can see that the two equations are almost the same. This is because those two strains are really identical apart from their labeling, B1 and B2. And the only two processes that are accounted for in the model is local growth. Here we chose the logistic growth term, and diffusion. Now, the only really thing I want to mention here is that you might notice that the diffusion coefficient is density dependent and decreases with increasing cell density. And this was really motivated by an experimental observation that we made, illustrated here by this image. We observed that if you start off with two initially separated colonies that are allowed to grow and so come closer to each other, once they meet, they don't really merge. Instead, they just abut, but there's no real merging behavior to be observed. So you get this somewhat smooth, really clear boundary between the two populations. And this motivated the use of this density-dependent diffusion coefficient. The other thing you need to know about the model is, of course, the spatial model is the spatial domain. We just use the disk to represent a Petri dish. Now, one more important ingredient for a mathematical model like this is the initial state, the initial condition of the model. And to choose or make an appropriate choice there, we again went back to the experiments and had a look at how actually the experimental system looks like early on. So on top here, you can see an image that was taken very early on during biofilm growth. Well, here it's not really a biofilm at this stage. But essentially what you can see here, and I'm not quite sure how well you actually can see the purple color here, but you definitely see the green spots here. You can see those separated microcolonies that are all of one single color, green and purple. And so essentially what has happened here is that every single one of those microcolonies can be assumed that they started off from a single cell that was able to grow and divide to form these microcolonies without any interference from the other strain. And that's why you get these single microcolonies with just one color in them. And so essentially in the mathematical model, we followed a very similar approach. This is just a schematic here, but essentially we chose a smaller disk in the center of our big disk, the inoculum of the biofilm, usually placed in the center of the Petri dish, and we at random placed these little microcolonies here. And like in the experiment, we always use the 1-to-1 ratio between the two, but obviously in the model, we can easily modulate the number of these microcolonies that we place. So that's our parameter in the model that how we can change the founder density. So that's the model setup. And so then we use those both approaches to compare the differences between different founder densities. And we just started off with some test cases. Here you can see 3 test cases each, one for low founder density, one for high founder density. And essentially we observed two important differences between the founder densities. The first one was no real surprise because that was already known from previous literature. But again, let me just repeat it, was that low founder densities induce spatial segregation. Okay, but the second important difference was that in the low founder density regime, even though we always started off with a 1-to-1 ratio between the two strains, we saw high variability in what we call competitive outcome. So simply defined as the relative abundance of one of the strains. And we saw this high variability across both the model and the experiment. So obviously that prompted us to do a bit of further investigation on where this variability was coming from. And this is really where the power of the mathematical model comes in, because if you observe variability, one sort of brute force approach is to just repeat the experiment many different times. And so what we observed by doing this, well, the power of the model is that obviously the model is much easier to scale. So there we were able to perform 1,000 replicates for each founder density, whereas in the experiment we had to restrict ourselves to just 12. But essentially in both cases, we saw this increase in the variability of competitive outcome for decreasing founder densities. And when I say increase in variability, I mean, here you can see that for low founder densities, really any competitive outcome was observed, which still was not very satisfying. So again, we really wanted to get to the bottom of this. And to do this, we came up with this very simple hypothesis that if you take the inoculum, take your microcolonies that you have, the hypothesis was that not every single one of those microcolonies contributes equally to the radial expansion of the biofilm. And the radial expansion bit here is key, because we hypothesized that only those patches that have this so-called access to free space can make a big contribution. So for example, that green microcolony here that's surrounded by others, our assumption was, or hypothesis was, that though that colony would not be able to make a big contribution. And we also quantified this hypothesis by drawing this big circle here, centered at the center of mass of the initial population. And then we color-coded it depending on which of the initial microcolonies is closest. So here it would be that purple patch, here it would be that green colony here. Then we defined the access to free space score of a strain, let's say the purple strain, to be the total length of all the purple segments relative to the whole circumference of the circle. And so our hypothesis then reduced to simply that this access to free space score should be correlated with the outcome. And so obviously we then applied this hypothesis to both the experimental and the modeling data. And here you can see 3 examples of the model of the experimental approach, where we applied this method here to early images and then compared it to the eventual outcome. And already here you see the suggestion that there's good agreement. And here you can see the data set of the modeling data for one low founder density, and you see excellent correlation. There's a slight complication here that if you change the founder density, you still have correlation, but the slope of the relation changes quite drastically. But this, at the same time, gives us an explanation of why there is so much increased variability for low founder densities. And I realize I just got 1 minute left or so. So let me very briefly say something about an extension to arbitrary strain pairs that, for example, interact with each other antagonistically. So we extended the model, and we also extended, I don't go into detail here, we also extended the experimental approach by combining the 2 strains. The important thing there was that both in the model and the experiments, there was one strain that was clearly stronger in killing the other than vice versa, always the green strain in both cases. And essentially what we saw is that at high founder density, the intrinsically stronger strain always outcompeted the other strain. So clearly at high founder density, the antagonistic interactions were the dominant mode of competition, but at low founder density, we again saw coexistence through segregation and large variability, even though we always started with a 1-to-1 ratio. And so the final question was that whether this predictor that we use for isogenic strains was still able to make good predictions, and the answer here was yes. Now, I'm going to just skip over the slides about the spatial heterogeneities. If you want to know about how this can be extended to a spatially heterogeneous environment, just talk to me or have a look at the slides. They're on my website. But I'm happy to talk about this later. But let me just finish by saying that essentially what we observed is that large variability in the competitive outcome in biofilms can be observed in biofilms inoculated at low founder densities. And not just that, we were also able to find the source of that variability. We identified the source to be the randomness in the spatial positionings, or the spatial distribution of microcolonies in the inoculum. And importantly, we found that this even is true if strains interact with each other antagonistically. And so to just finish by coming back to this initial motivational example of Bacillus subtilis as a potential biofertilizer. So essentially what that means is that if you're a company that is modifying Bacillus subtilis strains to sell them as biofertilizers, essentially you need to make sure that they both spread fast and also kill efficiently or fast for your biofertilizer to be successful, because depending on the founder density, different modes of competition are the dominant ones. So to finish, just a big thank you to the whole lab that I'm working in at the moment, the funders, and in particular all the people involved in that project, especially Margarita Calamara, who is a biology PhD student and who trained me to do the experiments, which was probably quite a difficult task. So, and also thank you to you for listening.
Thank you very much, Lukas, for a really nice talk. I see that Orkun's got his hand up.
I was wondering about motility, to think about how
So the question, the question was about how motility changes the situation. So, well, in the model, you know, it's almost like Fisher's equation, where the whole spatial spread really depends on both the growth term, the local growth term, and the diffusion term. So I would not expect there to be a real difference in the model, but in the experimental setup, we haven't considered this at all.
Yes, please.
It's interesting. I'm also working in biofilm, but more in drinking water. I'm wondering, is there any reason for you to choose certain type of soil? Because you mentioned garden soil. And the other thing I was thinking, do you think is there any difference if you're using different type of soil? Because like in Scotland, we have peatland, which is this carbon storage, and also methane, which is peatland soil.
Okay, so the question was about whether the type of soil makes a big difference. Well, the short answer is I don't know, because all the work that we did was just in the lab in a spatially homogeneous environment. That's where the spatial heterogeneity then comes in. But essentially, yes, I would expect the type of soil to make a big difference, because obviously it affects the nutrients and much more than that. The reason why we use the strain that comes from garden soil simply was it comes from a public engagement activity where we invited the public to essentially bring us a small soil sample from somewhere. Doesn't matter from where, but most of them obviously then go into their garden because they want to know what sort of bacteria live in their garden. And that's why. So we have quite a large library of Bacillus subtilis strains, but most of them come from garden soils simply because they were supplied by the public.
Okay, a quick question from Tom.
In the real soil, there could be quite a bit of predation. How would death affect your observations?
So the question is about how death by, let's say, other organisms, predators, affects situation. Again, it's not something we've looked at. However, we would argue that, so one thing that's important to know is if you go out here, take a soil sample, you will find Bacillus subtilis in there, most likely. So there's clearly a niche for Bacillus subtilis to exist in the soil. So even if you introduce a new strain, like a biocontrol agent, it's quite reasonable to assume that the sort of competitive pressure on the strain from other species will be tolerable, because Bacillus subtilis has a niche. It's just about the question whether one single new strain can persist among all the existing Bacillus subtilis strains.
Okay, thanks very much. Let's thank Lukas again. And our next speaker will be Daniel Garza from Leuven.